So, good day everyone. We are now in the third unit of this course entitled Genetic Engineering and Biotechnology that deliberately tackles about molecular genetic analysis and biotechnology itself. As this encompass a broad spectrum, what we will delve as for today is mainly about recombinant DNA technology. But before introducing to you the specific topics comprised by this discussion, it is important that you first know what is recombinant DNA technology. Recombinant DNA technology is determined by the Encyclopedia Britannica as the joining together of DNA molecules from different organisms and inserting it into a host organism to produce new genetic combinations that are of value to science, medicine, agriculture, and industry. Now, under this topic, I, Teacher Grace, together with my colleagues Teacher Britalyn, Teacher Cherry, Teacher Fatima, Teacher Teresa, and Teacher Rainali, will respectively discuss to you about cutting, joining, viewing, and locating DNA fragments, cloning genes, amplifying DNA fragments, DNA sequencing, and lastly, common methods used such as gel electrophoresis, southern blotting, PCR, and microarray. Let's start our discussion of recombinant DNA technology by considering two important tools used to construct and amplify recombinant DNA molecules, which are the DNA cutting enzymes called restriction enzymes and the DNA cloning vectors. These tools was largely responsible for advancing the field of molecular biology because a wide range of laboratory techniques are based on recombinant DNA technology. Recombinant DNA technology was made possible by the discovery of proteins called restriction enzymes or restriction endonuclease, which cut DNA at specific sequences, producing fragments that can be joined with other DNA fragments to form recombinant DNA molecules. Recombinant DNA molecules are DNA created by joining together pieces of DNA from different sources. With that being said, we will learn more about how DNA fragments are cut, joined together, viewed, and located for the first topic. In the year 1963, there are two enzymes isolated which are responsible for restricting the growth of bacteriophage in E. coli. The first enzyme is capable of adding methyl groups to DNA, while the second one can cut DNA, which they call as restriction endonuclease. Restriction endonuclease is specifically an enzyme produced primarily by bacteria that has the ability to cleave or cut DNA molecules at or around a certain base sequence. It is also called restriction enzymes, which serves as defense mechanism of bacteria against bacteriophage. So kaning si restriction enzymes class, it always cut DNA molecules at a particular point by recognizing a specific nucleotide sequence for 4 to 6 or 8 and more base pairs. This specific base sequence is known as recognition sequence or restriction site. Hence, recognition sequence or restriction site is the specific base sequence where restriction enzyme recognizes and binds to DNA. Recognition sequences exhibit a form of symmetry described as palindrome. The nucleotide sequence reads the same on both strands of the DNA when read in the 5' to 3' direction. Each restriction enzyme functions by inspecting the length of DNA sequence and recognizing a specific palindromic nucleotide sequences in DNA. Palindrome means a group of letters that form same words when read both backward and forward as illustrated in the picture. The enzyme then cuts both strands of the DNA within that sequence by cleaving the phosphodiester backbone of DNA, which refers as digestion of DNA. Each restriction enzyme recognizes its particular recognition sequence and cuts the DNA in a characteristic cleavage pattern. Hence, DNA molecules are cleaved in two methods by the restriction enzyme. First, it cuts both strands of DNA at exactly opposite points to each other. This results in DNA fragments with blunt ends, where two strands end at the same point. In other words, enzymes such as ALU1 cuts both strands at the same nucleotide pair, producing DNA fragments with double-stranded ends called blunt end fragments. So the second is common in most enzymes. It cuts the two strands of DNA double helix at different locations. This generates protruding ends. For example, one strand of DNA double helix extends some bases beyond the other. Since the target site is palindromic in nature, the protruding ends generated by such a cleavage have complementary base sequence. As a result, they readily pair with each other, and such ends are called cohesive ends or sticky ends. In other words, this creates offset cuts in DNA strands, therefore producing fragments with single-stranded overhanging ends. So, we have here a short animation of DNA cutting. What type of ends will this restriction enzyme produce? If your answer is cohesive ends or sticky ends, then you're absolutely right. Next up is how DNA fragments are joined. What we have here are two DNA fragments cleavage with the same restriction enzyme, which is the ACOR1, one of the first restriction enzymes to be isolated. These two DNA fragments come from different sources. Since both DNA fragments have complementary sticky ends, this allows them to anneal, or meaning to recombine. During the process, enzyme DNA ligase creates phosphodiester linkages between these fragments, which leads to the completion of the recombinant DNA molecule. For elaboration, the stickiness of ends facilitates the action of enzyme DNA ligase. When cut by the same DNA ligase, the resultant DNA fragments have the same kind of sticky ends, and these can be joined together by using DNA ligases. This property of restriction enzyme is of great value for the construction of recombinant DNA. Here is a short clip animating the role of enzyme DNA ligase in creating DNA recombinant. For the point to discuss in topic 1 is viewing DNA fragments. Gel electrophoresis is a method of viewing DNA fragments as well as in separating DNA molecules based on their size and electrical charge. It determines whether the restriction enzymes cut the DNA and the number and sizes of resulting fragments. This will be tackled deeper in later topics, so let's proceed to the last point of topic 1, which is locating DNA fragments. Southern, northern, and western blotting are the techniques used in locating DNA fragments. Southern blotting is a technique for detecting a specific DNA sequence in blood or tissue sample in the laboratory. The sample will be determined to contain the probe sequence if the probe binds to the membrane. Southern blotting is named after Edwin M. Southern. Northern blotting, on the other hand, involves transferring RNA from a gel to a solid medium. It is a technique for detecting individual RNA molecules and a mixture of RNA molecules. It is used for analyzing RNA from a specific tissue or cell type in order to determine gene expression levels. Lastly, for Western blotting, it is a scientific technique used to detect specific protein molecules in a protein mixture in which all of the proteins associated with a specific tissue or cell type can be found. It involves protein transfer from a gel to a membrane. The probe in this case is commonly an antibody, which is used to determine the size of a protein and its expression pattern.
Good day everyone, I am teacher Nino and today I am going to discuss about cloning genes. So what is gene cloning? It is the insertion of a fragment of DNA carrying a gene into a cloning vector and subsequent propagation of recombinant DNA molecules into many copies. Cloning can provide a pure sample of an individual gene separated from all other genes that it normally shares the cell with. Genetic engineering is the process of cloning genes into new organisms or altering a genetic sequence to change the product. Cloning vector is a stable, replicating DNA molecule into which a foreign DNA fragment can be inserted for introduction into a cell. So an effective Cloning vector has three important characteristics. So the first characteristic is an origin of replication, which ensures that the vector is replicated within the cell. The second characteristic is the selectable markers, which enable any cells containing the vector to be selected or identified. The third characteristic is the one or more unique restriction sites into which a DNA fragment can be inserted. So those are the three important characteristics of an effective cloning vector. Next is the steps of DNA cloning. So there are three steps in DNA cloning. So the first step is the cutting and pasting DNA. So cut open the plasmid and paste in the gene. This process relies on restriction enzymes and DNA ligase. Restriction enzymes cuts DNA while DNA ligase joins DNA. So our goal here in cloning is to insert a target gene into a plasmid. So the target gene here is the insulin gene. So using a carefully chosen restriction enzyme, we digest the plasmid which has a single cut site and the target gene fragment which has a cut site near each end. And then we combine the fragments with DNA ligase which links them to make a recombinant plasmid containing the gene. So that is the first step of DNA cloning. So the next step is the bacterial transformation and selection. Plasmids and other DNA can be introduced into bacteria such as the harmless E. coli used in labs in a process called transformation. During transformation, specially prepared bacterial cells are given a shock such as high temperature that encourages them to take up foreign DNA. So the next step is the protein production. Once we have found a bacterial colony with the right plasmid, we can grow a large culture of plasmid-bearing bacteria, and then we give the bacteria a chemical signal that instructs them to make the target protein. So that is what happens in the protein production. So those are the three steps of cloning DNA. So here is the fundamental steps in gene cloning. So the first step is the identification and isolation of the desired gene or DNA fragment to be cloned. Next is the insertion of the isolated gene into a suitable vector. Then after the insertion of isolated gene in a suitable vector, it is introduced into a suitable organisms or cell called host. So, the the vector multiplies within the host cell, producing numerous identical copies not only itself but also the gene it carries. During the division of the host cell, it copies the recombinant DNA molecules that are passed to the progeny and further vector replication takes place. After a large number of cell divisions, a clone of identical host cells is produced. So those are the fundamental steps in gene cloning. So next is the plasmid vectors. So plasmid vectors are circular DNA molecules that exist naturally in bacteria. They are commonly used as vectors for cloning DNA fragments in bacteria. It contains origins of replication and are therefore able to replicate independently of the bacterial chromosome. So as shown in the figure, the easiest method for inserting a particular DNA sequence into a plasmid vector is to cut its foreign DNA that contains DNA fragment of interest in the plasmid with the same restriction enzyme. If the restriction enzyme makes staggered cuts in the DNA, complementary sticky ends are produced in the foreign DNA and the plasmid. The DNA and plasmid are then mixed together, and then DNA ligase is used to seal the nicks in the sugar phosphate backbone, creating a recombinant plasmid that contains the foreign DNA fragment. Next is the transformation. Once a DNA fragment of the interest has been placed inside the plasmid must be introduced into bacterial cells. So transformation is the mechanism by which bacterial cells takes up DNA from the external environment. So some types of cells undergo transformation naturally, while others must be treated chemically or physically before they will go into transformation. Inside the cells, the plasmid replicate and multiply, and the cells themselves multiply, producing many copies of the introduced gene. Next is the screening cells for recombinant plasmids. Cells bearing recombinant plasmids can be detected by using the selectable markers on the plasmid. Plasmids make ideal cloning vectors but can hold only DNA fragments less than about 15 kilobase in size. When large DNA fragments are inserted into a plasmid vector, the plasmid becomes unstable and lose DNA. So as seen in the figure, a common way to screen cells for the presence of a recombinant plasmid is to use a plasmid with a copy of LAC-Z gene as a vector. The LAC-Z contains a series of unique restriction sites at which a piece of DNA can be inserted. So when a foreign DNA is inserted into the restriction site, it disrupts the LAC-Z gene and beta-galactosidase is not produced. So bacteria that are LAC-Z are exposed to the plasmids and then plated on medium that consists ampicillin. So last is the other cloning vectors. So these are the other cloning vectors: bacteriophage lambda plasmids, bacterial artificial chromosomes. Bacteriophage lambda infects E. coli, can be used to clone up to 23 BP of foreign DNA. It transfers DNA into bacterial cells with high efficiency. Plasmids are plasmids that are packaged into empty viral protein coats and transferred to bacteria by viral infection. So other cloning vector is the bacterial artificial chromosomes. So these are vectors that originally constructed from the F plasmid, which is a special plasmid that controls mating and the transfer of genetic material in some bacteria. And it can hold very large fragments of DNA that can be as long as 300,000 BP. So those are the other cloning vectors. So let us now proceed to the next topic.
Amplifying DNA fragments. Amplification is a process to produce multiple copies of specific DNA sequence. DNA amplification technique. The most common method used in DNA amplification and the most well-known technique in molecular biology is the polymerase chain reaction or PCR. It is a biochemical process capable of amplifying a single DNA molecule into millions of copies in a short time. PCR was developed by Kary Mullis in 1983. These are the following PCR components: template, primer pairs, DNTPs, DNA polymerase, and cofactors and buffer. As for the template, this is the sample of nucleic acids that contain the desired target sequence and is separated initially into two strands in PCR reactions. As for the primer pairs, primers are short oligonucleotides or synthetic short DNA strands which complement the target sequence and bind to the single-stranded DNA. For the DNTPs, DNA polymerase requires deoxynucleotide triphosphate, also known as DNTPs. The nucleic acid components or nucleotide bases adenine, thymine, cytosine, and guanine, or the ATCG, to generate new DNA. As for the DNA polymerase is a thermostable enzyme that synthesizes copies of DNA with every consecutive cycle of PCR. Polymerases are responsible for binding free nucleotides complementary to the template DNA. And for the cofactors and buffer, magnesium or Mg2 plus is an important cofactor for DNA polymerase. Magnesium assists phosphodiester bond formation and is required for successful PCR amplification. Buffer solution, providing a suitable chemical environment for optimum activity and stability of the DNA polymerase. Now let's go to the three major steps of PCR technique. These are the denaturation step, annealing, extension or ligation step. Denaturation step. This step is the first regular cycling event and consists of heating the reaction to 94 to 98 degrees Celsius. It causes DNA melting of the DNA template by disrupting the hydrogen bonds between complementary bases, yielding single-stranded DNA molecules. So the double-stranded DNA is separated into two single strands by breaking the hydrogen bonds between the nucleotide base pairs. The next step is the annealing step. This is the next stage of the PCR process. In this step, primers anneal or bind at complementary sequences of the denatured DNA strands. The reaction temperature is lowered to 50 to 65 degrees Celsius for 20 to 40 seconds, allowing annealing of the primers to the single-stranded DNA template. Lastly is the extension or elongation step. Extension is the final stage of a cycle and occurs at a temperature that allows optimal polymerase activity of binding nucleotides to the annealing primer, resulting in exponential amplification of the template strand. The temperature at this step depends on the DNA polymerase used. Taq polymerase has its own optimum activity temperature at 75 to 80 degrees Celsius, and commonly a temperature of 72 degrees Celsius is used with this enzyme. At this step, the DNA polymerase synthesizes a new DNA strand complementary to the DNA template strand. By adding DNTPs that are complementary to the template in the 5' to 3' end direction, condensing the 5' phosphate group of the DNTPs with a 3' hydroxyl group at the end of the extending DNA strand. After elongation, the cycle goes back to step 1, usually for 20 to 40 cycles. In this diagram, we can see that in the first cycle, there is 2 copies of DNA being produced after going through denaturation, annealing, and elongation. In the next cycle, the second cycle, it increases into 4 copies and increases into 8 copies in the third cycle. Under optimum conditions, if there are no limitations due to limiting substrates or reagents, at each extension step the amount of DNA target is doubled, leading to exponential amplification of specific DNA fragment, hence the amplification of multiple copies of specific DNA sequence. Next in line is the advantages and limitations of PCR. As for the advantages, PCR is the preferred method for application-oriented fields involving nucleic acid amplification for its simplicity, easier methodology, extensively validated standard operating procedure, and availability of reagents and equipments. As for the limitations, PCR cannot be used to amplify unknown targets. Prior information about the target sequence is necessary to design the primers. DNA polymerases are prone to error, which potentially causes mutations in PCR products. PCR is very sensitive to contaminations. A small amount of contaminating DNA could result in misleading or ambiguous results. So, the limitations in the PCR technique gave birth to alternative methods. The alternative nucleic acid amplification methods are the following.
Alright, so let's talk about DNA sequencing. DNA sequencing is defined as the process of determining the sequence of nucleotides in a piece of DNA. Sequencing allows the genetic information in DNA to be read, providing an enormous amount of information about gene structure and function. This picture here shows what a DNA sequence looks like. So let's now proceed to the first generation sequencing technologies. In the mid-1970s, Frederick Sanger and his colleagues created the dideoxy sequencing method, or simply called Sanger method. Picture below shows the process of Sanger sequencing, which we will tackle in detail later. And at about the same time, a second method called the Maxam-Gilbert method was created by Alan M. Maxam and Walter Gilbert. So after Frederick Sanger created the Sanger method, it quickly became the standard procedure for sequencing any purified fragment of DNA. It is also important to note that this method of DNA sequencing is based on replication. The fragment to be sequenced is used as a template to make a series of new DNA molecules. In the process, replication is sometimes terminated when a specific base is encountered, producing DNA strands of different lengths, each of which ends in the same base. So what do we need to conduct Sanger sequencing? So here are the ingredients for Sanger sequencing. It includes a DNA polymerase enzyme to synthesize the DNA. Another one is a primer, which is a short piece of single-stranded DNA that binds to the template DNA and acts as a starter for the polymerase. The next one is the 4 types of deoxyribonucleoside triphosphates. However, that is not all. Sanger sequencing actually has a unique ingredient, which is the dideoxyribonucleoside triphosphate. So, dideoxynucleotide are similar to regular or deoxynucleotide. However, there is one key difference, which is that they lack a hydroxyl group on the 3' carbon of the sugar ring. What does this imply? So in a regular nucleotide, just like here in figure A, this hydroxyl group actually acts as a hook, allowing a new nucleotide to be added to an existing chain. And since the dideoxynucleotide lacks this, No further nucleotides can be added and the chain ends with the dideoxynucleotide. So here is the method of Sanger sequencing. So as we can see here, there is a template, primer, and DNA polymerase which are combined in a tube. So each tube, one type of dideoxyribonucleoside triphosphate is added. So after that, when a dideoxynucleotide is incorporated into the growing chain, synthesis terminates because the dideoxynucleotide lacks 3' hydroxyl, which I explained. So synthesis terminates at different positions on different strands, which generates a set of DNA fragments of various lengths, each ending in a dideoxyribonucleotide with the same base. So after that, the fragments produced in each reaction are separated by gel electrophoresis. And the sequence can be read directly from the bands that appear on the autoradiograph of the gel, starting from the bottom. The sequence obtained is the complement of the original template strand. After the first generation DNA sequencing technology, another method of DNA sequencing technology were created, and they were more efficient and less expensive than the Sanger sequencing method. The next generation sequencing techniques include pyrosequencing and Illumina sequencing. So if Sanger method is based on DNA replication, pyrosequencing is actually based on DNA synthesis. Nucleotides are added one at a time in the order specified by template DNA. The addition of a particular nucleotide is detected with a flash of light, which is generated as the nucleotide is added. So here is the method of pyrosequencing. The DNA to be sequenced is fragmented. Adapters containing primer sequences are attached to each fragment. The DNA is made single-stranded. After that, each DNA fragment is attached to a bead surrounded by a drop of solution containing reagents of PCR. The bead actually used to hold the DNA and later to deposit it on a plate for the sequencing reaction. The DNA fragment is amplified by the PCR. After amplification, DNA is made single-stranded and each bead is forced into a well and layered with sequencing reagents. So, nasulod na sila sa well, DNA synthesis takes place na dayon. A solution containing a specific deoxynucleoside triphosphate flows over the plate. And then when a nucleotide is added to the growing chain, pyrophosphate is released and produces a light-emitting chemical reaction. So the light is measured by an optical detector. And then the amount of light is proportional to the number of nucleotides added. So if the template in a well specifies 3 successive adenine nucleotides, then 3 nucleotides are added and then 3 times more light is emitted. Now let's proceed to Illumina sequencing, which is a little similar to Sanger method. Special nucleotides are used that have a fluorescent tag attached with a different colored tag for each type of nucleotide. Each nucleotide also has a chemical group or a terminator that once incorporated into the growing DNA chain, it prevents the incorporation of any additional nucleotides. So it will stop, just like what the dideoxynucleotides do in Sanger method. In this case, however, unlike the Sanger method, the terminator is reversible. Moving on to the third generation sequencing technology. If we think that second generation sequencing technology is already efficient, then the third generation sequencing technology is even more advanced and rapid. Under this is nanopore sequencing, which can determine the sequence of a single molecule of DNA. In this method, a single strand of DNA is passed through a tiny hole in a membrane. So, tiny hole, which is why it is called nanopore. And so the reason why third generation sequencing technology is being developed is because they wanted to be able to sequence an entire human genome for less than $1,000. So that inexpensive.
There are 4 common methods used in detecting and viewing DNA fragments. First one is gel electrophoresis, second southern blotting, third PCR, and lastly microarray. In order for us to know more of these methods, I'll be discussing to you first the gel electrophoresis. What is gel electrophoresis? Gel electrophoresis is a technique commonly used in laboratories to separate charged molecules like DNA, RNA, and proteins based on their size and charge. On the right side, you can see a sample image of a gel electrophoresis. The movement of charged molecules in this method is called migration, and agarose gels are typically used to visualize fragments of DNA. These gels are made up of polysaccharide called agarose. Next, how is gel electrophoresis carried out? In order to perform this method, the first thing we need to do is to prepare the gel. So there are two options you can choose. The first one is to make the gel or to use pre-made gels. For you to make the gel, you need to have an agarose powder and mix it with an electrophoresis buffer and heat it to a high temperature until all of the agarose powder has melted. After you have prepared the gel is then placed into the main body of electrophoresis, which you can see on the sample image on the right side. Here, after you place the gel, you can now pour the electrophoresis buffer solution into the tank until the surface of the gel is covered. For preparing the DNA for electrophoresis, the first thing you need to do is to add a dye to the sample of DNA to increase the viscosity of the sample, which will prevent it from floating out of the wells and so that the migration of the sample through the gel can be seen. Once it is done, each of the DNA samples you want to examine is carefully transferred into one of the wells. A DNA marker, also known as a size standard or a DNA ladder, is loaded into the first well of the gel. You can observe on the first image below that the wells are located near on the negative pole, while the second image illustrates the placement of loaded DNA fragments and agarose gel in buffer. The next step in this process is to turn on the gel bags, and current begins to flow through the gel. Also, we need to remember that DNA molecules have a negative charge, so they start moving through the matrix of the gel towards the positive pole. So right now, we will observe the second image wherein you may notice that the bands are separated. So here, the longest DNA fragments migrate most slowly through the gel and stay closest to the wells, while the shortest DNA fragments migrate most quickly through the gel and away from the wells. In this case, the fragments are now separated by size. To further our knowledge, we will proceed to the visualizing of the DNA fragments. So once the fragments have been separated, we can examine the gel and see what sizes of bands are found on it. When a gel is stained with a DNA binding dye and placed under UV light, the DNA fragments will glow, allowing us to see the DNA present at different locations along the length of the gel. The glowing DNA fragments can be seen on the image on the left side. A well-defined line of DNA on a gel is called a band. Each band contains a large number of DNA fragments of the same size that have all traveled as a group to the same position. By comparing the bands in a sample to the DNA ladder, we can determine their approximate sizes. For instance, the bright band on the gel above is roughly 700 base pairs or BP in size. So the glowing DNA fragments on the left side is roughly 700 base pairs in size. Since we are already done with gel electrophoresis, we will now proceed to southern blotting. Southern blotting is a laboratory technique used to detect a specific DNA sequence in a blood or tissue sample. It is designed to locate a particular sequence of DNA within a complex mixture. For example, southern blotting could be used to locate a particular gene within an entire genome. After we define and give meaning to the purpose of southern blotting, we will now proceed to its method. Here on the image, there are 6 procedures needed to perform. The first one is to digest the DNA with an appropriate restriction enzyme. This enzyme cut a DNA molecule at a particular place. Second, run the digest on an agarose gel. Third, denature the DNA. Fourth, transfer the denatured DNA to the membrane. Fifth, probe the membrane with labeled ssDNA. This is also known as hybridization. Lastly, visualize your radioactively labeled target sequence.
What is PCR? PCR is a technique used in the lab to make millions of copies of a particular section of DNA. It was first developed in the 1980s. The polymerase chain reaction, or PCR, was originally developed in 1983 by the American biochemist Carey Mullis. He was awarded the Nobel Prize in Chemistry in 1993 for his pioneering work. PCR is used in molecular biology to make many copies or amplify small sections of DNA or a gene. Using PCR, it is possible to generate thousands to millions of copies of a particular section of DNA from a very small amount of DNA. PCR is a common tool used in medical and biological research labs. It is used in the early stages of processing DNA for sequencing, for detecting the presence or absence of a gene to help identify pathogens during infection, and when generating forensic DNA profiles from tiny samples of DNA. Principles of PCR: thermal cycling, selective unrepeated application with the help of primers, Taq polymerase isolated from Thermus aquaticus. As the reaction progresses, the DNA generated is used as a template. How does PCR work? The principles behind every PCR, whatever the sample of DNA, are the same. There are 5 ingredients to set up a PCR, and those are DNA template or target DNA, primers, DNA nucleotide bases, Taq polymerase, and buffer solution. However, in some sources, kay nagamit pa sila laing requirement, which is magnesium chloride salt solution. There are 3 main stages of PCR, and those are denaturing, annealing, and extending. When the double-stranded template DNA is heated to separate into 2 single strands, that is denaturing. When the temperature is lowered to enable the DNA primers to attach to the template DNA, that is annealing. When the temperature is raised to the new strand of DNA is made by the Taq polymerase enzyme, that is extending. Next, we will go to the clinical applications of PCR. Role in diagnosis of infectious diseases, role in cancer diagnostics, genetic diseases, and paternity testing. Next is variations and modifications of basic PCR technique, that is, reverse transcription, quantitative PCR, nested PCR, asymmetric PCR, and multiplex PCR. What is microarray? DNA microarrays are solid support, usually of glass or silicon, upon which DNA is attached in an organized grid fashion. Each spot of DNA, called a probe, represents a single gene. History: microarray technology evolved from southern blotting, where fragmented DNA is attached to a substrate and then probed with a known DNA sequence. The use of miniaturized microarrays for gene expression profiling was first reported in 1995, and a complete eukaryote genome on a microarray was published in 1997. Principle: the principle of DNA microarray lies on the hybridization between the nucleotide. Using this technology, the presence of one genomic or cDNA sequence is in 1 million or more sequences can be screened in a single hybridization. The property of complementary nucleic acid sequence is to specifically pair with each other by forming hydrogen bonds between complementary nucleotide base pairs. Steps in DNA microarray. First, sample preparation. Second, purification. Third, reverse transcription. Fourth, labeling. Fifth, hybridization. Sixth, scanning. Seven, normalization and analysis. There are two types of DNA chips: the cDNA-based microarray, an innovative technology that facilitates the analysis of expression of thousands of genes simultaneously. Next is oligonucleotide-based microarray. The probes are short sequences designed to match parts of the sequence of known or predicted open reading frames. The DNA chips are used in many areas as given below: gene expression profiling, discovery of drugs, diagnostics and genetic engineering, alternatives, splicing detection, proteomics, functional genomics, DNA sequencing, and lastly, toxicological research. Thank you.